Hi, A&Pers. This is Dr. Jones checking in. This is A&P1 Lab 4. So this is our neuro lab, and let's start going through some of our neuro models. First is the neuron model, and if you're following along in your book, we're on page, for me, 31. So the neuron model, a neuron is another word for a nerve cell. The dendrites are these gray things sticking out, and they receive information. The soma is the cell body, so the soma, and in the center is the nucleus, and you can see the nucleoli in the nucleus. The perikaryon is the part of the soma that is not the nucleus. So imagine the donut without the hole, except three-dimensionally. So maybe the donut without the jelly. There we go. So the donut without the jelly is the perikaryon. The axon hillock is the orange triangle, and that's where your information processing takes place, and that connects the soma to the axon. And here you can see the axon continues. This part is axon dissected open, still axon, more axon, and then you can see that the axon is covered. So this layering that covers the axon, the whole layer, is the myelin sheath. And each bit here is a Schwann cell. So if the axon were a hot dog, then the Schwann cell is the tortilla or crepe or pancake or whatever it is you use that's wrapped around your breakfast sausage here. So each tortilla is a Schwann cell, and the layer of tortillas is the myelin sheath. The gaps between individual tortillas or Schwann cells is the node of Ranvier, capital R, it was the guy's name. And the endoneurium, just like the endomysium was a rough connective tissue covering, the endoneurium is a rough connective tissue covering. Instead of myo for muscle, now we have neuro for nerve. So endoneurium surrounds and electrically isolates each of the axons. The neurolemma is the outer membrane of each Schwann cell. So if this were a pancake wrapped around the breakfast sausage, because all of this are layers and layers of Schwann cell because it's one pancake wrapped around and around and around. But it's just this outer edge of it that you could pour syrup on that is the neurolemma. The axolemma then is just the cell membrane of the axon. So structure here is axon, membrane is axolemma. Layer here is myelin sheath, cell, Schwann cell, membrane, neurolemma. And then the indent here is a node of Ranvier. Rough connective tissue covering is the endoneurium. Okay, so we haven't covered these things, and they look a whole lot like this. So when the axon splits to go to individual synaptic knobs, the splits that go to the individual synaptic knobs are called a telodendrium. So kind of like your hand splits into fingers to ends at your fingertips, your axon splits into telodendria to end at synaptic knobs. So each synaptic knob, you can see the mitochondria in there, each synaptic knob contains these pearls called synaptic vesicles, and each vesicle contains, general term here, neurotransmitter. Now the neurotransmitter that we talked about most in the muscle system was acetylcholine, but that's only one kind of neurotransmitters. There's lots of different neurotransmitters, and these synaptic vesicles will carry one. Each cell only carries one different type of neurotransmitter. So that brings us back to this little guy. So this is a telodendrium of another neuron come to communicate with this neuron. So you can see the synaptic knob. They've even got some little pearls in there for synaptic vesicles. Cool, right? Let's see if we can make it focus. There we go. Okay. So the membrane of the synaptic knob that releases the neurotransmitter via exocytosis is going to be your presynaptic membrane. And remember, these synaptic knobs, axon terminals, synaptic terminals, whichever you're calling them, don't actually touch. There's a gap there between the synaptic knob and the next cell, and that gap Okay, yep, there's no gap here. I'm having to hold it apart. But that gap that I'm creating here is the synaptic cleft. And in this case, this plastic here will be the postsynaptic membrane. The postsynaptic membrane will be the membrane with the neurotransmitter receptors. Okay, so that's the neuron models. We'll call that video 1, AMP1, lab 3. There you go, it's a nice shot. Video 1. The education must go on. Dr. J out.